Alright class, this video is the review for Tuesday's exam. It will be up to this afternoon on Sunday. Please take any questions you have to the Piazza page. I've been getting really good responses and really good questions from you guys on there. It's also very helpful that a lot of your classmates are answering those questions before I get to them, because either I'm in class or I'm driving home, since I drive an hour and 15 minutes away from campus to come home. So keep it up, that's great. All of your classmates' answers so far have been correct. I go back and I verify if they're correct, or if there's a slight change, I'll mention it. But other than that, take it to the Piazza page. Please remember Dr. Kokash's review session is going to be tomorrow. I believe it's in East Hall, but reference her email about it that was sent about a week ago. On this exam, there's going to be, looks like 1, 2, 3, 4, 5, 6, 7 chapters. One of them, Dr. Kokesh had you guys review on your own, which was chemical principles. That should be review for most of you guys, since I know the requirement is 2 bio classes before this. It was mainly on macromolecules, but I'll go over that in a little bit of detail in this class, in this review. I do want to mention this is not going to be a step-by-step of the study guide. The study guide is for you guys to do on your own, because if I just gave you the answers to all of the questions on the study guide, it would not help, because I'm going to tell you right now, the study guide is not the exam. It's going to help you on the exam, but it is not the exam. So please don't just study the study guide. Please go over the PowerPoint she gave you. They're going to be your best bet. Also, if you have a few questions, small questions about, the mechanism of this, I got a question on Piazza, NAD plus, how does it go to NADH? I love those questions. I'll be happy to answer them, but the answer is also in the book. I know you see him in the back. This is Mosby. He's not going to move. He runs the house. He's king of this. As you can see, he's on the couch, and I'm on the floor. So you see how that goes. So I'm going to start with the study guide. The first question is kind of vague. It's about the individuals in microbiology. Please know them all. I know that's a lot to say, but this class is going to be memorization, head to toe memorization. Once we get to bacteria and viruses, all that, it's all memorization. So just start now. It's perfect. You're going to need all this if you're especially going into nursing. You're going to need to memorize a lot of stuff. This is a great class to begin that skill. So just some of the people to remember. I absolutely remember Pasteur. He was the one who invented the S-shaped flask. Please remember that. Please remember the reason the S-shaped flask was such an important invention at the time. Remember Joseph Lester. He's the one who described how to spray the operating rooms. And of course we use that now. It helped reduce the death and infection rate once you got out of surgery. Remember Robert Koch? He was a medical doctor. He's the one who did the germ theory. That's going to be big in this class, so please remember that. He also has his own postulates. Please just, look over those. Make sure you know them. Make sure the golden age, the dates of that, which is 1857 to 1914. Please remember those. That's when Pasteur did fermentation and pasteurization. It's when Lister did the aseptic surgery, which is the cleaning with phenol. Koch did the germ theories. The gram stains were presented, and the procedure was laid out, and the isolation of many disease-causing organisms were also laid out. Know who coined the term magic bullet. That would be Paul Ehrlich. Please remember that. That is a very important. Alexander Fleming. I think we all know what he did. We all know he is the inventor of penicillin, which penicillin is going to come back as well in this course. So just please remember who created it and penicillin, and get ready for that joyride once penicillin comes back. That's pretty much all that people to remember. Just go back over. I put that it starts on slide 31 of your introduction PowerPoint. Just please look back over it, do a little bit of memorization, remember who it is, and you'll be perfect for those questions. To the chemical principles PowerPoint. Dr. Kokash had you do this on your own because it is common knowledge you need coming into microbiology. It should came from the 2 courses that you took in biology before this course. Just know what the macromolecules are, which is carbohydrates, lipids, proteins, nucleic acids. Know the different kinds. Know the different carbohydrates, what they are. Carbohydrates are sugars. Then remember your monosaccharides, which is simple sugars. It can be 3 to 7. Remember a few of them: pentose, tetros, heptose, tritose. Just remember a few of those. Remember disaccharides are 2 monosaccharides linked together. Then we are going to remember polysaccharides. Just remember poly, 8 or more. I have seen in a few of my other biology classes polysaccharides go up to 100. So just remember 8 or more polysaccharides. Lipids, know what it is essential for. It is essential for the function of membranes. So remember that. Remember that they're, of course, fats are triglycerides. They're fatty long chains of hydrocarbons. Let's see what else you should remember from this. Definitely remember the hydrophobic and heterophobic regions, hydrophilic regions. Hydrophobic regions, of course, are the fatty acid tails. That's why when you look at a bilayer membrane, you see that the tails are inside because they are hydrophobic, they do not want to touch water, and then the heads are hydrophilic and they are on the outside. Remember saturated and unsaturated. Saturated is single chain, so they're just going to be in one nice single chain. Unsaturated is not. They're going to kink out, so they're always going to be the ones that kind of look like an awkward L shape, are the half of the K. Just some of our proteins. They're, of course, proteins. They're essential for the function of the cell. We cannot live without proteins. Just know that they can be enzymes. Hopefully you guys know what enzymes are. Just remember, it's common knowledge. Nucleic acids. Hopefully we all know that's DNA, RNA. Just remember DNA, double, RNA, single. See what else I want you guys to know about this. I think that's good for that one. Let's move on to question 6 on your study guide. Virulence factors. Know what virulence factors are. Also know what capsules are. Know the functions of a virulence factor. So of course it's going to be able to attach to surfaces. It's going to protect from phagocytosis, and it can also be eaten as food if it gets to that level. Question 7 is important. Please look at question 7. Question 7 is going to be major. It is the list 8 differences between prokaryotes and eukaryotes. That will be on the exam. So I don't know how much more to stress that. It's going to be on it. Know pretty much everything. Listing 8 would be great, and I would memorize those. Just remember, eukaryotes are larger, prokaryotes are not. Remember, eukaryotes have a true nucleus, and they have membrane-bound organelles. Prokaryotes does not have a true nucleus, and they do not have membrane-bound organelles. Remember, eukaryotes 80S ribosome, prokaryotes 70S ribosome. Remember, eukaryotes have a simple cell wall. Prokaryotes have a cell wall with peptidoglycan. And just remember the main one, one of the main ones. Remember, eukaryotes replicate by mitosis, and prokaryotes replicate by binary fission. Also, this is a major question on your study guide. Please know it. List the differences between gram-positive bacteria and gram-negative bacteria. Some of the main ones I would know: peptidoglycan layer in a gram-positive is thick, and in a gram-negative it is thin. The acids, the teichoic acids, please remember that they're present in gram-positive, not positive in gram-negative. That's going to be hints towards the questions. Dr. Kulkash can write a question and say, blah, 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 contains teichoic acids. You're obviously going to know. And then the options are going to be gram-positive, gram-negative, both or neither. It can be a mixture of those. But, of course, you know teichoic acids. Wait, they're present in gram-positive. It can't be gram-negative. Obviously, it can't be both. And I do remember that they're in gram-positive, so it can be neither. So question is A. That is how you need to focus on this exam. It's not tricky if you know the material, because it gives hints in the questions. Like, it will be a long question, but one sentence will be, has a thick peptidoglycan layer. You're going to be, wait a minute, gram-positive has a thick peptidoglycan layer, and that is going to be the answer. So just please know the material. I know it seems hectic, and I know it seems very intensive, but the more you know it, and the more you go in there confident knowing it, you're going to do way better on the exam. The other questions: flagella, what is it used for? Locomotion. Sex pili, remember what the sex pili does? It's where bacteria uses their sex pili to attach to another bacteria to transfer material. Know what an endotoxin is. Another big question, what are organelles? So if you go to slide 14 in the eukaryotic cells PowerPoint, it's going to give you a good breakdown. It's going to start with a list: nucleus, endoplasmic reticulum, Golgi apparatus, mitochondria, chloroplast, lysosomes, vacuoles, and centrioles. Please remember them. This is going to be a 50-question exam, so Dr. Kokash can give you 3 of them, or she can give you 1 of them, because there's going to be an A exam and a B exam. So you never know which one you're going to get, so it'd be best just to know them all. So I'll just give you guys a few quick materials and facts that you need to know. The nucleus, of course, is the largest internal structure. It's oval in shape. It holds the DNA that we need. And then the cell divides, of course, mitosis and meiosis in the nucleus. There's a great picture on slide 16. It breaks down the nucleus itself. Endoplasmic reticulum, are the a cytoplasmic membranous network structure. Just know some of the functions. It synthesizes lipids and proteins. And of course, there's rough and there's smooth. Know what is part of them, so know that the rough has attached ribosomes. That's what makes it rough. It's like when you get chill bumps, you rub your hand across your skin. Of course, there's going to be bumps. It's going to be rough. Smooth, just it's no ribosomes. You know, just think you put lotion on your skin. It's smooth. I don't know. I just remember stuff like that. So if that works for you, please do it. If it's silly, hey, silly might get you an A. So do whatever helps you know the material. Golgi apparatus. So they're found near the nucleus. They're of course the discs. I always remember them as pancakes. Because they literally resemble pancakes to me. And just know it receives and transports lipids and proteins. So when I first learned this in my biology class in high school, I knew them as a UPS system because it transports and it receives. Transports and it receives. That's pretty much what the Golgi apparatus or complex does. Mitochondria, we all know, powerhouse of the cell. Nothing's going to work without mitochondria. Of course, cellular respiration, energy production, ATP. So there is a question on Piazza that I got, how energy is created for ATP. I put a great answer, cellular respiration. You know, it starts with mitochondria because, once again, powerhouse of the cell. The next one is chloroplast. We all know what chloroplasts are. It's the site of photosynthesis in plants, and it replicates autonomously. Don't know if I said that right. Hope I did. Main thing, though, of course, site of photosynthesis. Lysosomes contains hydraulic enzymes. So you will see this a lot in the course. Right now, just remember it's derived from the Golgi apparatus. It's important for phagocytosis, and it's found actually in large numbers of white blood cells. So if one of the questions says, where is lysosomes found, just know it is found largely in white blood cells. And of course, the main function, important for phagocytosis. Vacuoles, it is a cavity space for storage. Once again, I know it as the trash can. You dump stuff in there you want to store or throw away, and you put it aside. Once again, this is derived from the Golgi apparatus. So remember, two things that are derived from the Golgi apparatus, of course vacuoles, and then of course it's going to be, the lysosomes, both derived from the Golgi apparatus. The next question on the study guide I felt was really, really important. I have seen this on every exam I've either seen or I've taken myself in microbiology, especially the lab. It always seems to show up in the lab. What are plasmids? So if you learn it now, better for you in the future. Plasmids, I'll give you the exact definition, are extra chromosomal double-stranded circular DNA that carries genes that are often not essential for survival. So just think it's extra chromosomal material, mainly a circle, double-stranded, of course, don't forget that, double-stranded, and it just kind of floats around, and it contains material that we actually don't need for survival. We have it, it's great, it's like, I don't know, I'm looking at a cover here on my chair. I never use it, but it's not essential for me, but I have it, so it's there. So just remember, plasmids carry stuff that's not important. Endospores. No, endospores are resistant bodies produced by some bacteria. So resistant as in it does not go away. I know Dr. Kokesh on the first day talked to you about the anthrax attack that happened at the post office. The reason it took so long for that post office to reopen was because the endospores left from the anthrax could not be killed. They just were not being killed by anything that was tried and used. So, of course, the little other material was killed, but the endospores remained. So they had to work and work and work because they're resistant. Classification, know the binomial nomenclature. That's how they are classified in our day and age. Please remember who actually started that. It was, I will tell you the background of him. It was in the 18th century. It was Carolus Linnaeus. It was the first nomenclature. So he did not do the binomial nomenclature, but he did the first ever system to classify something, because right before him we just, oh, this is this, this is this. He actually, his life's work was to create a book, all in Latin of course, that caused of a genus and a species name, so we'd be able. So one of them was H. sapiens, or K. pneumoniae. So just know there's a genus and there's a species. Always know the genus comes first, the species is next. So Homo sapiens, the genus is Homo, the species is sapiens. The next, what are the 5 kingdoms? Please remember, it is plants, fungi, animal, protista, and then Monera. Please just remember those 5. Please know what's inside of them. So please know plants, of course, are plants. Fungi, of course, are fungi. Animals are animals. And then know protista and Monera. Please also know that that is different from Wussi's domains, which are eukarya, bacteria, and archaea. They are 2 different domain kingdoms. What's another big question? Viruses. How are viruses classified? That is a trick question. This appears on a lot of exams. Viruses are not classified because they are not cells. So just know, if you get a question that says, oh, What kingdom would our domain would viruses be classified in? No, it's a trick question. The answer is none because they are not cells. I got a couple questions on Piazza about oxidation reduction reactions. A way I learned it in biology and chemistry was a quick mnemonic: oil rig. Oil is oxidation is losing, rig reduction is gaining electrons. It's just a quick, simple mnemonic. If you ever have trouble, is it losing, is it gaining, to just remember that mnemonic. I also got a couple questions about the differences between catabolism and anabolism. To be honest, I just had to keep cramming this into my brain. It was a difficult topic for me to learn too at first, so I just kept studying. I will give you guys the exact definition so you guys can remember it, and hopefully you can make up a silly mnemonic or a way that you remember it. Anabolism is the buildup of complex organic molecules from simpler ones. And involves the dehydration, synthesis, or the removal of water. Of course, like you're dehydrated, you have no water, and are endergonic. Catabolism is the breakdown of complex organic molecules into simpler ones. And of course, this is done by hydraulic reactions, and it's exergonic. So once again, I just have to put them in little lists. Anabolism, the buildup of complex using the dehydration, and it is endergonic. And then catabolism is the breakdown into simpler molecules. It is using the hydraulic, and it is exergonic. I have yet to come up with a mnemonic for that. I just have to keep pounding it in my mind until I remember it. Unfortunately, you will come across a few of those things in this class that you will just have to keep studying to remember, and unfortunately that is one of mine. Other than that, microbial growth is a big one. Please remember the calculation. If you are struggling, go to Khan Academy, please. Khan Academy is going to be your best friend for this. All you do is you type in on Khan Academy, you type in microbial growth calculation, and it will pop up. Please remember the calculation. I do not know if Dr. Kokesh is going to give it to you. So please remember, it is MF, which is final, equals M and I, our initial, times 2 to the N. Please remember that. Please know that N is the number of generations. So it could be 2 generations, 5 generations, 10 generations. It's whatever Dr. Kokesh feels on putting on the exam. Please note MI is the initial number of bacteria, and MF is the final number of bacteria that you have once the initial has gone through many of the different generations. Like I said, Khan Academy is perfect. They are very well scripted, and they're very well in actually breaking down the material to better help you with this calculation. Please don't think that is the only thing that you're going to get on the microbial growth PowerPoint. I've had a few people ask me that. Is this going to be the only material? And no. The calculation is a big part, but please don't remember, you have very other important things in this PowerPoint. Phases of growth is a big one. Please remember not only how to draw the chart, but also how to label it. Say Dr. Kokesh already gives you the graph, and it's of course got the flat line, the increase, the plateau, and the down. Please remember the phases. I'll just tell you now, it's lag, log, stationary because it plateaus, and then death because it decreases. Please remember that. Please know the generation are double timing. That once again plays a fact into your calculation. Also remember the growth requirements, obligate and facultative. The physical requirements you needed, which are pH, temperature, and osmotic pressure. Please remember the biochemical requirements: water, oxygen, organic factors, carbon, nitrogen. Please remember those. Know the 3 different temperature ranges. I know a lot of people are like, oh, I didn't even know it was going to be on the exam. It is. Please study it. The psychrophiles. once again, are cold loving. You're going to find those in the Antarctic Ocean, the Arctic Ocean. You're going to find those at the bottom of the ocean because it likes the cold. Mesophiles, moderate temperature loving. If you want to classify humans as something, moderate temperature. Remember, the temperature range is 10 to 47. Remember, thermophiles, heat loving, 40 to 80. Extreme thermophiles is 65 to 110. I have not seen Dr. Kokash use this question, but I received it in my advanced microbiology class. Where would you likely describe and categorize a bacteria that like to live in a hot spring? Once again, hot spring, you're going to know it's really hot temperature from 65 to 100. Of course, it's going to be extreme thermophile. Please remember 3 categories once again: psychrophiles, mesophiles, thermophiles. Please remember hypotonic, isotonic, and hypertonic. Isotonic, water moves out, solution moves in, it stays the same, no change. Hypertonic is where it's going to shrink. So there's more water in solution going out, so it's going to shrink. Hypotonic, concentration of solutions inside and outside, is going to bust. I know this was on your concept map. Know the difference between heterotrophs and autotrophs. Please know the difference between heterotrophs and autotrophs. Once again, you need carbon sources, nitrogen sources, sulfur surfaces. This is the biochemical requirements. The effects on oxygen for the growth. I've got a bunch of questions on Piazza about these as well. Obligate aerobes, facultative anaerobes, obligate anaerobes, aerotolerant aerobes, and then micro aerophiles. I can tell you right now, you might get a slide picture. This was slide 17 of your microbial growth PowerPoint. Please review that. Dr. Kokash could give you the test tubes, and it's going to have aerobes spread out, aerobes at the top, aerobes at the bottom, and then you're going to have to label which one are these. You will have to do that in BioLab. So for you guys in Bio 306, you will have to do this for one of your lab reports. So it is good to get familiarized with slide 17. Obligate aerobes, they're only aerobics, so you require oxygen to grow. Once again, that's obligate aerobes. Facultative anaerobes, both aerobic and anaerobic, it can grow in the presence, not in the presence of oxygen. It doesn't really matter, but it does grow greater in presence of oxygen. Obligate ceases in the presence of oxygen. Does not grow, stops. It comes to a halt. So aerotolerant aerobes, it continues in the presence of oxygen but really doesn't grow. And then micro aerophiles are oxygen required in low concentrations for them to grow. So please, if you look at the test tubes, obligate anaerobes, they're all normally at the top for oxygen. Facultative, you got some at the top, some at the bottom, some in the middle. And once again, it can grow either at the top or the bottom. Obligate anaerobes, at the very bottom. It does not want oxygen. It wants to be deep in the solution. Aerotolerant, mainly at the bottom, but you have a few at the top. And then micro aerophiles, it is not at the very top, not at the very bottom, but a little higher than medium because it wants low concentration of oxygen. It does not want to be consumed with oxygen, just, dipped. Know what biofilms are? Just know that there's how microbes live in communities. They gather together. I don't think Dr. Kokash has it in the slides, but there is a picture of a tongue that I've seen. Then, to be honest, even if you brush your tongue, you're still going to have biofilms of bacteria living on your tongue. So just know biofilms are communities, are clumps of bacteria that do live together, and they create a biofilm. So like I said, that's a lot of microbial growth. This is a very important PowerPoint. please look over this one. If you only have time to look over a few, I suggest highly that you look over microbial growth. The very last PowerPoint, microbial metabolism. Once again, catabolism, anabolism, definitely know those. Enzymes, know what they are, know what catalysts are, know what a holoenzyme, an apoenzyme, and a cofactor are. You're going to need to know those. Remember, apoenzyme is the protein component. The cofactor, or the coenzyme, is the non-protein component. So apoenzyme, protein, cofactor, non-protein, plus those together, that's how you get the holoenzyme. Let's see. Do know the mechanism of enzymatic action. Please know that. Let's see what else. Know the effects of pH and substrate concentration on enzyme activity. Once again, oxidation and reduction. Oil rig. Oxidation is losing electrons. Reduction is gaining electrons. Substrate level phosphorylation. Of course, it's the direct transfer of PO4 to ADP. No, oxidative phosphorylation is using the electron transport chain. Of course, phosphorylation is the photosynthetic of cells. Glycolysis. Absolutely, no glycolysis. It's the splitting of sugars. The best I can tell you is please study slides 35 and 36. Your best friend. Please study, once again, 35, 36, 37, 38, and 39 of the microbial metabolism PowerPoint. It will help you immensely when it comes to glycolysis questions. I do not know, once again, I've not seen the exam. I do not know how many questions are going to be on here. I'm just trying to help as best possible what I've seen in the past, and glycolysis has been on. Definitely know electron transport chains. Chemoosmosis is the mechanism by which ATP is generated during the electron transport system. Please know the protein gradient. And then fermentation. Once again, fermentation, what it's used for: cheese, yogurt, beer, and please know who created it, which was Pasteur. I'm almost positive I'm not saying his name right, but you know who I'm talking about. Louis Pasteur created the S-shaped flask, pasteurization, and fermentation. Please remember that. So that is everything that I believe is important. Like I said, I have not seen the exam, so please study all 7, I believe. 2, 4, 7. All 7 PowerPoints. Please study them. Please do the study guide. The study guide will help immensely. Even though it is not your exam, it won't be question for question. There will be some things that's not on the study guide that will be on the exam, and that's why I say please, please, please study the exam. Study the PowerPoints and the study guide for the exam. Like Dr. Kokash said at the beginning of class, this is going to be one of the hardest classes you're going to have. It is going to be extremely difficult, extremely time-consuming, extremely intensive when it comes to memorization. Once we get closer to the bacteria and the viruses, I'm going to make a video laying out how I study for them, because you're going to need a lot of memorization. But at that point, put that to the side and memorize this. I would love to say you can dump this material after, but you cannot. This will be on the final exam, so please study for it now. The more you study for it now, the better you'll do on your exam, and the better you will do on the final exam, because you already know it. All you have to do is flip through it really quick, and you won't have to stress yourself out at final exams, because unfortunately you might have another exam that day, another exam the next day, because final exams are in such a short period of time. Quick reminders: Scantron. Please bring a Scantron. Green or blue. I have seen more green than blue, but it doesn't matter. Just make sure there's no marks on it, and make sure it is not creased. I know people throw their Scantrons into their book bag and they just put their books on top because they might be rushing up the house that morning, and it ends up creasing the Scantron. The biology Scantron reader will not read it, so please try and nicely and neatly put your Scantrons in your book bag the morning of. Please bring a pencil. Pins is just too risky. With microbiology, you might think it's an answer and get so stressed out for time, and then you realize you did really well on time, and you go back and you're like, oh man, wrong answer. Please just bring a pencil. Number 2 pencil, mechanical pencil, just a pencil, please bring it. Like I said, know your microbial growth calculation, because I do not know if Dr. Kokesh is going to give you the equation. Remember, it's MF, which is the microbes final, equals MI, which is microbes initial, plus 2N, and the N is the generations. Have one by, it could be 2, 5, 10, whatever Dr. Kokesh decides. Thank you guys for watching this video. I hope it helped some of you at some point. I know you guys are stressing. Y'all will do fine if you just make sure you study the PowerPoints, you study the study guide. I hope to goodness you guys have already started studying. But please tonight and please tomorrow, all the time you get, study this. Like I said, ask me questions on Piazza. I'm always happy to help. I always check it at least 3 times a day. And I know your classmates are very good on having my back, answering your guys' questions. I will always come back through and I will say, yes, this is correct. Oh, there's a few things changed, or whatever. Also, if you have any questions that either was arised due to this video or due to your studying, Dr. Kokesh is having a review session in East Hall, I believe, tomorrow, but please check the email she sent about a week and a half ago. Thank you guys for watching the video, and I will see you all on Tuesday. Good luck studying.